My interest in ancient DNA started when I joined Svante Pääbo's lab in Germany to do a postdoc. Soon after I got there, he told me that the work that I had signed up to do, coming over there to look at gene expression in chimpanzees and humans, that I wouldn't be doing that. I would be working instead on a different project to analyze DNA from Neanderthal. And we're going to sequence the whole genome of the Neanderthal, and you're going to do it. And I said, okay. I'm Ed Green, professor of biomolecular engineering at University of California, Santa Cruz. It was the first time anybody had used this high-throughput sequencing to look at all of the DNA that you can get out of bones that are tens of thousands of years old.
Our genome can't be understood without comparing it to the genomes of other species. But the key prize was the species that is most closely related to us, and that is the Neanderthal, an extinct species that is our closest genetic relative. And a superstar in that group was Ed Green. Ed Green exploded onto the scientific scene with the first genome of a Neanderthal. That's when I recruited him to Santa Cruz.
I came to Santa Cruz in 2010. My research group here at UC Santa Cruz has been working on this really big main technical challenge in ancient DNA, which is efficiently converting the small amounts of fragmented DNA that you can recover from an old bone, for example, into the form that you put on a DNA sequencer. This technique is useful for ancient DNA, but it's also useful for health and forensics. And this technique we call the Santa Cruz reaction. Santa Cruz is a fantastic place to be an entrepreneur. I have started 3 companies. I like both things. I love the academic research that I do in my lab. It has shifted over the years toward more and more technology development. I've learned that that is where my heart is. And for whatever reason, I think I tend to, attract people into the lab who are also interested in technology development. Brandon Rice was a fantastically talented grad student who was in my lab at the time. He was, a few years into his PhD and volunteered, threw his name in the hat to be CEO of this company. And before I could blink, really, there was a company that had been started called Dovetail Genomics that was generated to use this idea to provide this service for folks who need to sequence a genome, and it was a game changer.
So basically, what was done for the Human Genome Project for humans, for all the other plants and animals and critters that we did, lobsters and humpback whales and axolotl, and you can't do anything like that without a reference genome. And so we were building reference genomes. And we did our 50 or 75 reference genomes that we built. Each one of those unlocked a whole area of research on that organism. That's hundreds or thousands of publications or lines of research that we enabled.
And another company, Australia, that he spun off, actually looks for small pieces of DNA from crime scenes. So Ed became the person to go to when you had a DNA sample from a crime scene that no other technology could get any information from.
This was a market that was announcing itself to me, and it's a very uncomfortable home to be doing that kind of thing in an academic lab. So that was a company that was built out of necessity. The DNA that we can get from hair, for identifying human remains, the DNA that comes out of those bones, it's like ancient DNA, just not quite as ancient. Every sample that came, I thought it would be the last one, and at some point it became obvious there wasn't going to be a last one.
Since 2017, the DNA Doe Project has been using investigative genetic genealogy to solve unidentified human remains cases. One of the specific instances where an organization like the DNA Doe Project would turn to Australia and Dr. Green would be an instance where the only sample we have would be rootless hair. In 2000, the bodies of 2 unidentified victims were found in a barrel in Bear Brook State Park in New Hampshire. In 2024, the case was brought to us at the DNA Doe Project. The Estrella team was able to produce a much stronger set of bioinformatics for us. And finally, we were able to identify this victim as Rhea Rasmussen, who now, after many, many years, we've finally been able to give her name back. Dr. Green is truly a friend to the investigative genetic genealogy community.
It's meaningful work in a way that really nothing I've ever done is meaningful. It matters to people. Here at Santa Cruz, I'm the scientific director of QB3. Design, implement, carry out programs that are designed to nurture the entrepreneurial ecosystem around biotech. And it's a consortium, 3 campuses, UC Santa Cruz, UC San Francisco, and UC Berkeley. We also do workforce development, have programs for undergrad students in the summer to take an internship at a biotechnology company or a technology company in general.
Ed has a wealth of experience in successfully starting bioscience companies that bridge the gap between research in the lab and real-world impact. That's an invaluable perspective for leading students at QB3 who want to do the same.
My main expectation is that there will be a new Ed idea and a new company, and then a few years after that, yet a new Ed idea and a new company.
It's been great doing all this technology development in DNA sequencing and in genomics in UC Santa Cruz, which is really, one of the preeminent places in the world for genome development.